Good morning, everybody. My name is Manali Mukherjee. I'm welcoming you warmly to this much anticipated webinar from the International Eosinophil Society. And today's webinar is on mucus plugs in asthma and the role of eosinophilia and airway obstruction. I am an associate professor at McMaster University with a lot of interest on this topic, and of course a self-proclaimed eosinophilophile. And hopefully at the end of this webinar we will come to understand where eosinophilic diseases and how mucus occlusions may be well intertwined and leading to clinical manifestations. Co-moderating, co-chairing this webinar is Andrew Higham. He's a budding translational scientist at the Division of Immunology at University of Manchester, and he is, as he calls, my mucus buddy, because he has a lot of interest in this as well. Since I chair the early career committee at IES, Andrew is a very active member, so I'm very happy to co-chair this webinar with Andrew. I would like to thank our, I also want to give a little bit of briefing about IES. It's an organization of scientists and clinicians. We are all interested in eosinophil. It's a blood cell that we know is strongly associated with many diseases. It's very difficult to work with, but we love working with it. We love the challenge. The society sponsors biennial meetings. The recent one was in Montpellier. There are monthly webinars and other educational programs for our 300 staggering members, many of them who are early careers. We review new information about eosinophils and its role in health and disease. We have given a QR code here. Please do consider joining IES, and definitely a shout out to the trainees. Do join. This is a wonderful space for trainees and to really get the space to collaborate and network. And we have our 14th biennial Congress in Vancouver for 2027, July. We're all very excited. Please save the date, and we'll see you in the beautiful city of Vancouver. IES Biennial Symposium is coming back to Canada, so please save the date. And before we start with our first speaker, I would like to thank Sanofi Regeneron for very generously sponsoring of this webinar and making this happen. Thank you, Sanofi and Regeneron. Right, so just some housekeeping. Please use the question and answer button to ask any questions. You can do that anytime during the presentation, and both Andrew and I will read the questions to the speaker at the end of their talk. All attendees are muted, and the chat feature is disabled, so it's the Q&A that you can use to ask questions. This webinar is being recorded and will be posted on the IES website. There is closed captioning or live transcription during webinar, so please, you can click the show captions button in the Zoom format. Right, so we have a fantastic speaker lineup. We will start the webinar with Dr. Christopher Evans speaking to us about the emerging cell and molecular targets for treating mucus hypersecretion in asthma, followed by Dr. Burton Dickey. He'll be talking to us about agonist-stimulated mucin secretion in airway mucus occlusions. And finally, Dr. John Fahy, who will be putting all of this together and bringing our favorite cell back into the picture, talking about eosinophils and mucus in fatal asthma. So without further ado, I believe we have 108 participants right now online. We do have about 200 registered, but considering the 3 lineups and the talk and the discussion that I'm anticipating that will happen, we'll start it. So Dr. Christopher Evans is a professor of medicine at University of Colorado, and I remember reading all his papers on MUC-EC5 and MUC5B and all the mucins when I had to educate myself on understanding what mucus is all about. So here's Dr. Evans speaking to us more about the different cellular and molecular targets for targeting mucus hypersecretion. Dr. Evans, it's all yours.
replace the current share. We're good there. Okay. Good. I hope I'm sharing the appropriate screen.
Yes.
Okay, good, good. I didn't see the little green outline. So thank you very much for this invitation. I was initially concerned that I wasn't going to have enough to talk about with eosinophils in a long lecture. So I was glad to see that a couple other people were included in here that are better mucus experts than I am. And I'll do the best I can to help open up some discussions that will come along the way. So I'm going to just start by mentioning my only disclosure here is I'm a consultant for a company that's related to pulmonary fibrosis, but does relate to Muc5B in that sense, but it doesn't affect any of the information I'm going to share today. So I thought I'd start by outlining a little bit of the definitions here that might help as we go through the rest of the talk here, but also the other two lectures today, which is just put two simple things into place. First is when we talk about mucus, we're talking about a gel, a polymeric hydrogel that sits on the top of the airway surfaces. So this is what lines mucosal tissues. It's comprised of proteins, the backbone of which is these things here called mucin glycoproteins. So I'm going to try to be very careful about saying mucin, talking about the protein, and mucus only refers to the extracellular material, with one caveat that mucus, O-U-S, is the adjective that kind of blends between the two. But for the most part, when we talk about the mucin proteins themselves, because they're the important constituents that really give mucus hydrogels their gel forming and transport and barrier properties. The two predominant ones in the airways are Muc5AC and Muc5B. They're very large proteins, so each monomer is about 500 nanometers long. They form dimers in the ER. They progress through the Golgi and become polymerized, where they can become up to about 30 microns long, and at the same time also take on massive amounts of glycosylation, which cause them to increase their molecular masses by about 5-fold during that process. So you take something that's very, very big and make it ginormous, and that creates a lot of work for a mucus cell to do. And so that's where we really want to focus on how mucins are synthesized and what their functions are once they get outside the cells. So the main thing that mucus does on the airway surface is that it provides a protective barrier. In the airways, it sits on top of epithelial cells, and ciliated cells that are found beneath the mucus layer are responsible for allowing transport of the material in a steady state. This can be enhanced by things like cough, which help us to propel phlegm globbers faster than a mucociliary transport machine can do on its own. But that escalator is very important in the steady state, and diseases in which you lose it are ones that can have devastating effects on the on airway clearance and lead to infections. The situation that occurs in asthma, though, is one in which the amount of mucus that's being produced and the levels of secretion that occur are such that they become overwhelming. And in a fatal cases of asthma, this feature here of mucus plugging is something that's almost universally seen. And this has been noted for almost over a century now, but has been carefully dissected by pathologists and more molecular readouts over the last several years. So the important part here is that mucus hypersecretion is an important phenotype, a detrimental phenotype in disease that's related to the poor control of expression and secretion of the mucins Muc5AC and Muc5B. To investigate this, we predominantly in my lab worked in mouse models in order to be able to work with these very large proteins that you can't just throw in a plasmid and express in CHO cells and play with them in vitro. You have to actually do quite a bit of work to engineer mice or cells in ways that will allow you to study the full-length polymers. We started this by making knockout mice. The first study I'm going to show here is studies using Muc5AC knockout mice. And the rationale behind that is that the healthy versus allergic asthma-like state in the mouse, which is characterized by mucus metaplasia, seen here with PAS staining, is one that's characterized by the induction of Muc5AC in the epithelium. And the importance of Muc5AC in this setting is such that if we knock it out, we can reduce airway hyperresponsiveness. So methacholine responses, in this case in a inhaled methacholine in a mouse model, you can see that there's a decrease in the changes in resistance that occur in the hyperreactive mouse in a knockout setting. So it's this pink allergic line here. And that protection is related to a prevention of mucus plugging in the airways, which we've shown here and we quantitated in that paper. So since then, this has made us really start to focus a little bit deeper into mechanistic questions about how it works. And when I think about how mucus works, I think about two sides of this. It's sort of the production side of things, where you have gene expression, protein biosynthesis, and post-translational modifications, and then secretion. And I think that the importance I've sort of exaggerated here is where we've spent most of our time focusing is in these middle steps here in the biosynthesis pathway. As I alluded to before, mucins are big proteins. So at a scale level, when you compare them to something like CCSP or SPC, they're the blue whale next to the scuba diver here in terms of the mass of material. And it's, again, just at the protein level, and you haven't even added in the polymerization and glycosylation steps. Cells are confronted with a big problem in just dealing with the size. And they're actually pretty complex. So this is sort of a molecular model of what I just showed you before, in which I'm depicting here the different folds and three-dimensional, in a two-dimensional setting here, model of the three-dimensional pieces that need to come together to make a mucin. So it's got these conserved von Willebrand domains in the N-terminus, and in the C-terminus is also von Willebrand factor-like domains as well. These are important for polymerization of the mucins, and they're what really holds the material together and allows the polymers to have viscoelastic properties. The central part between them is unstructured and is where all the proline, threonine, and serine rich domains are that are the sites of O-glycosylation. And again, that's really what gives mucins their defining glycoprotein characteristics. But the process of building these involves about 100 to 150 disulfides per molecule. And again, that's as they polymerize, become a massive workload in the ER and the Golgi. So in the process of producing them and really focusing in on the disulfide bonds that are formed, we focus in two areas. One is in the ER, which is in the first steps of synthesis here, where the C-terminal regions of the mucin proteins form dimers. These protomers are then transported into the Golgi, where they polymerize at their N-termini, and the N-terminal disulfide domains that are formed kind of mature over time as the proteins are being glycosylated, and they take these pairs of dimers and turn them into the full-length polymers. So when we wanted to see whether or not these were important, we asked a simple question, which is, what if you get rid of the disulfides? And we did this on a wide scale by using a reducing agent called TSEP. And we took mice that were allergen challenged, we put them on a ventilator, and as we were measuring methacholine responsiveness, in this case to IV methacholine, we had them inhale TSEP at various points along the way. And this prevented airway hyperresponsiveness. So the TSEP treated animals are here in pink, and you can see here that the airway hyperresponsiveness occurs in saline mice, saline vehicle treated mice that are, again, all allergen challenged, are protected against in the mice that receive TSEP. And that protection occurred with the elimination of mucus plugging in the airways, which I'm depicting here, and we quantified as well. So this is having justified the disulfides as an important area of functionality in the mucins, we've decided to go deeper into this at a molecular level. And in this respect, what we've done is taken advantage of what's known in the field of von Willebrand factor biology. So von Willebrand factor is actually an evolutionary descendant of mucins. So mucins came first, and then von Willebrand factor evolved from them. But the field is a bit farther advanced in terms of where they're at with their structural biology. But you can take these guys and line them up, and we can find it at the C-termini of the mucin. So this here, there's a CK or cysteine knot domain. There's 11 cysteines that are conserved across all of them. So von Willebrand factor, MUC5AC, and MUC5B in both human and mouse. They all line up, and I'm highlighting these guys here that I call 5, 6, and 11 based on their order in this domain. And their importance is shown here in a crystal structure of human von Willebrand factor, where they're involved in the formation of disulfide bonds. So number 11 pairs up with another 11 on another subunit, and 5 and 6 kind of crisscross each other and create a structural subunit that way. So we took and modeled this in AlphaFold using, in this case here, I'm showing mouse MUC5AC, and we were able to produce a structure that looked very similar to what was found up here, which provided us some evidence that this is worth pursuing and considering as a potential area to look at for mechanisms of disulfide assembly in the ER. So we're going to focus in here on C5, 6, and 11. And what we did was we took mice and we used CRISPR to gene edit them. Again, as I said earlier, this is such a big, massive protein that you can't just throw it in a plasmid and put it in cells and do anything with it. And so for us, it was actually easier to make gene-edited mice and to take and use CRISPR to replace the cysteines in those various sites in order. So I'm showing them here as C5, 6, and 11, where we change cysteine to serine, which is a structurally similar, but it's a hydroxyl instead of a thiol group, and it doesn't form disulfides, but it kind of has the same shape. And what we found is if we knocked out number 5 and 6, we actually lost the protein, which is interesting in its own way. But 5, 6, and 11 mutation created a situation in which only dimers were produced. So I'm showing here is a Western blot gel. This is agarose because these things are, again, so big that they actually don't get into STS page. And what you see up here is the polymers at the top end. And then at the bottom end here is in this non-reducing setting is monomers and dimers that kind of fall out. As a control over here, we've partially reduced some material with dithiothreatol, where you can see monomers and dimers here. And what we found again is that the 5-6 mutation, this kind of internal piece in here, where 11 was still intact, created a situation in which the protein was misfolded or somehow detected as being aberrantly produced, and the cells. I'll show in a second that it looks like the cells are degrading it. But the 5, 6, 11 was secreted, and it was secreted as dimers as we had expected or hypothesized based on what was known with von Willebrand factor. And so we're looking at this as a potential means for creating a mucus preventive phenotype, right? So you can structurally go in and attack a specific mucin molecule, in this case MUC5AC, potentially at these sites that are found within it. So as I alluded to before, that 5-6 mutation was one that was interesting, where we found that the material wasn't being secreted. It was being produced and retained in the ER, and we think degraded. That degradation occurred without a loss of MUC5B production in the same cells. So we think that it's not causing a toxic ER stress environment in the cells. And again, it could be considered as a potential therapeutic option down the road. But when we looked at the effects of these mutations on mucociliary transport, this is work that was done in vitro in collaboration with Esther Vladar here at University of Colorado. And let me see, just turn this off here. And what we were able to show in vitro is that if we put particles onto cells, in this case, these are wild type cells that are treated with IL-13, you can see that most of the cells are just kind of stuck. So they produce so much mucin that they overwhelm the mucociliary transport apparatus. But if we mutated that 5-6, where we really got rid of MUC5AC and there were protected transport, and the triple mutation with 5, 6, and 11 seemed to do something along the same lines. It was not quite as much as we saw in the sort of full knockout setting, which is what we're considering with that 5-6 mutation. We've begun to quantify these by doing particle tracking. And what you can see here is that there's a distribution where there's a lot of non-moving particles in the wild type, and that movement is improved in the mutants. And when we quantify even just the maximum velocity, so in the tracks that do move, we can see that there's an improvement in the velocity of the beads as they move through the mucus layer. When we look at this in an airway hyperactivity setting, as I had showed earlier with the mouse models, so these are mice that are on a flex event getting inhaled methacholine, we can see that the airway hyperactivity that occurs in wild type mice is prevented in the mutant animals. So in this case, the 5-6 and 5-6-11 mutant animals both show protection. So as we move forward with these, we're trying to find out more about the processes in which they're made. We also have additional work that's focusing more on the Golgi and the N-terminal disulfide assembly that occurs there, but I don't have data to present at this time on that model. But what I would like to do is to go a little bit farther into that next stage of something else that goes on in the Golgi, which is glycosylation. So I want to make sure I'm careful of time here. So the glycosylation steps that occur are such that you have in the cis Golgi, you have O-GalNAc being added on to serines and threonines. The additional sugars that get added include GLICNAc and galactose, these sort of circles and squares that are shown here. But eventually what happens is you get down to the business end here where you've capped them off with specialized proteins, in this case, fucose and sialic acid. And at sort of a broad level, there are two very different sugars here. Sialic acid is very charged and polar, whereas fucose is more hydrophobic. It's a denser molecule that potentially will retain less water. So we think that this is going to be something that creates a more viscous gel, and this is something that makes more lubricative gel. And it turns out that in the baseline state, this is the predominant species that is found in the mouse lung. And in an allergic setting, fucosylation is something that's induced. So we're tackling this by looking at how it gets induced, and we're using a enzyme that is pretty uniquely qualified as the fucosal transferase for mucins, and that's FUT2. And actually work that John Fahy published many years ago now showed that there's an association with FUT2 deficiency, or I should say FUT2 sufficiency, and severe asthma exacerbations. So preventing fucosylation may be a way to help reduce the asthma phenotype. In a deceased patient, we can see that the plugs that form are laden with MUC5AC, shown in magenta, and the overlay of UEA1, which is a lectin that detects fucosylation, is positive. So we can see here that there is quite a bit of dense fucosylated material in the airway. And we can see a similar phenotype in mice where fucosylation is something that's present in the goblet cells that are found in the airways after antigen challenge. When we take FUT2 knockout mice and expose them to allergen, we can find that, like the other models I've shown before, they're protected from airway hyperactivity. This protection occurs with a reduction in mucus occlusion and obstruction in the airways. And we think that this is a way that we could look at a preventative phenotype. So as we move forward with this, we actually just finally got a new grant activated to start looking at this at a molecular level, where we've taken and modeled FUT2 against published sequences and experimentally derived structures of human FUT9 and mouse FUT8. So similar fucosal transferases that are in different cell types and have different enzyme substrates in terms of their sugar structures. But the active sites are reasonably well modeled compared to FUT2. And we're able to take and model FUT2, and we identified amino acids and structural domains that are present within the donor and acceptor domains for the glycosyltransferase that could be involved in synthesis. So we're just beginning now to tackle these. And then also to tackle questions about how the fucosyltransferase enzyme itself works and how it gets localized to the Golgi. Long story short, there's things in the transmembrane and cytosolic domains that help it know to go to the trans Golgi, we think. And at the interface between them, these actually function as dimers. There's apparently pH-sensitive amino acids that may help and facilitate dimerization and enzyme function. So what I'd like to do is the very last point here is to come back to another question about how mucins work. And I promise I'm going to actually say eosinophil again here. And in this case, what I'm going to do is to talk a little bit more about what we think mucins are doing in the context of inflammation and tissue injury, as well as repair. So what I'd like to do is to highlight work that we did in collaboration with Bruce Bachner several years ago. And he's really the person who brought us into this field and brought me back to eosinophils from my from the glory days working with Dr. Fryer back in Baltimore. And the thing that was brought to our attention was that the sugars that are present on mucins, in this case sialic acid, with a focus here on MUC5B, but there's no reason why MUC5AC or other mucins could not have similar structures. The sugars that are present on them can be ligands for lectin-type receptors. So Bruce was focusing on cyclic 8 in the human and cyclic F in the mouse as eosinophil lectins that could be used to dampen eosinophilic inflammation. And we had an interest in collaboration with Bill Jansen here at National Jewish to look at macrophages. And I'm going to just show two final slides here where I'm going to allude to some data in which we've recently shown that the macrophage function is something that's of interest. And I think that what we're finding in macrophages could be applied to a chronic eosinophilic disease as well. So what we found is that this relationship between MUC5B and its sialic acids that are present on there, acting as ligands on cyclic F, are things that affect biology. In a bleomycin exposure model of pulmonary fibrosis, we found that the interaction here, at least the cyclic F role here, is one that's required for the fibrotic response in response to bleomycin. And we're still trying to figure out how that works mechanistically and what targets are involved. We have a recent follow-up study that's just been reviewed, and actually we just got our major revision response this morning from JCI Insight, showing that there's a little bit of an opposite response in terms of what happens with cyclic F deficient mice in a model of emphysema. So it looks like there's programming in the macrophage that's somehow regulated by cyclic F. But we don't really know how this happens. And where we're really starting to move towards here is to take a deep dive into what the mucins are doing beyond that glycan interaction and what other protein domains may be interacting as well. So the paper I showed earlier where they crystallized von Willebrand factor cysteine knots had a figure in it that showed a whole bunch of other cysteine knot related proteins in it. These include the TGF-beta family. And what I found to be very interesting in that sense is that we could take and draw some of the same shapes that they have with the cysteine knot from von Willebrand factor and maybe allude to something that's occurring with the TGF-beta family as well. And what we've proposed is that there may be an interaction in which you have coordinate signaling between a Siglec and a TGF-beta-mediated signal within leukocytes that affects their biology. Again, it's something we're pursuing, and at this point, as I believe Dave Jacoby has said before, is it's exciting data that's not encumbered by the presence of actual data yet. But we actually do have quite a bit of phenomenology that puts some proof in the pudding there. And so we've modeled this against a bunch of other TGF-beta-like family members, and we do think that there's reasons to pursue this going forward in terms of looking for receptor interactions and not necessarily all ligand matching per se. So my final summary here is just that we think that the disulfide bonds that are used to hold mucins together are things that are important for the mucus functions that we see at a clearance and plugging level, but we think that they're also really important for other interactions that they might have, potentially with eosinophils based on their proteins that are protein structures and domains that are present on the mucins and potential receptor families that could be interacting on the eosinophil. So with that, I'd just like to thank the people who did the work, and I hope I have not run too far over time.
Perfect timing. Thank you very much, Dr. Evans, for a fantastic start to this webinar. While we wait for some of the questions to come in, I really would encourage the early career members to ask questions today. It's a great opportunity to ask experts in the field about any burning questions you do have. So I really would encourage that. So I wait for some of the questions to come in, Dr. Evans. So in terms of the fucosylation and the role of that, is there any evidence of inflammatory endotypes having a role in that process? Perhaps it's more prevalent in type 2 high inflammation, or whether neutrophils also have a role in determining the fate?
Yeah, so what I can say is I think endotypes can be characterized in part, in this case, based on secretor status. So about 20% of European people are deficient in the FUT2 enzyme activity. So they are the secretor negative population. So there could be some balance in there. Now that means that 80% of people do fucosylate. So that's a big group for an endotype. But what we do find in mice and find that is a very interesting phenomenon is that when we take a naive mouse, there's essentially no fucosylation in the airway mucins. It's mostly MUC5B there, not MUC5AC, but I don't know that well, actually, we have evidence that it doesn't, the glycans don't really care which mucin it is. If we give a mouse LPS, we don't see fucosylation. If we give a mouse IL-13 or an allergen, we get tons of it. If we give them other bacterial challenges and other types of IL-1 or even type 17 type of challenges, we don't see that same thing. So it does look like the fucosylation pathway is something that is skewed towards type 2 immunity. And I think it makes sense because I think it probably relates to how mucins need to glom onto and bind to things like parasites. And in fact, in the gut, fucosylation is pretty common in the steady state, despite being a lot of bacteria present on MUC2 all the time. So there's a lot of programming and endotypes that could be teased out of that, but it's purely speculative.
Great, thank you very much.
Yeah.
So the first question is from Per Venge. So what do we know about patients with von Willebrand deficiency in terms of asthma and mucin production?
I know nothing, Jon Snow. Yeah, I don't really know. I mean, so von Willebrand disease and the hematologic disorders that kind of characterize that are things that I don't necessarily lump into the same area of this. And as a non-clinician, I have to admit, I don't really have a good answer for you. Sorry.
So the next question is from Helena Agata. So looking forward to the papers on Siglec F, how can you distinguish between the interaction with Siglec F on alveolar macrophages and eosinophils? Does this have the same effect in both cell types?
It does not. And actually, the initial pilot studies we were doing with Bruce about 10 or 15 years ago now, we actually compared macrophages and eosinophils. And macrophages, unlike eosinophils, which if you stimulate cyclic F on them, if Bruce can correct me if I'm wrong, I saw his name on the list here, it triggers the apoptosis quite profoundly. The alveolar macrophage is quite resistant to that. It's a very persistent cell type. So it doesn't seem to regulate apoptosis, and we don't really see much in terms of that paper, the two papers I showed there. We didn't see much in terms of programs that would allude to changes in cell cycle or cell death. It seems that it's more of a blunting of inflammatory response and maybe pro-reparative. And that's where we think that coordination with TGF-beta-like signaling may be something where you're inducing a repair mechanism, which is bad in a chronic lung fibrosis setting, but it's actually really important for repair, right? So if you take an epithelium and you disrupt it, right? So if you create tissue injury, the mucus is now coming into contact with the mesenchyme and also with the macrophages that are lining up near there. And having a coordinated signal that says, let's not spaz out with an inflammatory response, let's turn on some pro-repair genes and get cells to reorganize and fill in the gap is something that I think could be very enticing. Again, it's something that we're still in the process of studying.
Great, thank you.
Yeah, thank you.
So next one is, great talk, Chris. Is that inhaled therapy you use to decrease airway hyperresponsiveness in the mice able to work in humans, or is it toxic?
TSEP is nasty stuff, and I'm going to defer to Dr. Fahy, who I assume is going to be talking a bit about that later on today. But there is a effort to produce reducing agents that have that chemical effect without toxicity.
Okay, on to the next one. So we've probably got time for a couple. I think we're ahead of schedule. Time for a couple more. So how specific is FUT2, and is it a potential therapeutic target for antagonists in treating asthma?
So it's surprisingly specific, which is why we focused on it as a glycosyltransferase. We actually spent several years trying to pursue sialotransferases, and there's a lot of redundancy. So FUT2 and alpha-1,2 fucosylation of mucins is a really nice selective interaction. And that's exactly what we're going for in this new grant, where we're picking apart the amino acids that are present in the active site that allow for electron transfer and salt bridges to form and create that GTPase type of activity that's needed for adding the sugars onto the proteins. So how we can interact with those either at an allosteric or active site is specifically what we're going for in that. So let's talk in 4 years.
So we'll probably have two more questions now. So this one's from James Cornfield. Given the bioactive role you are hypothesizing that mucins can play in disease, do you anticipate any potential negative consequences to disrupting mucin production as an asthma therapy?
Yeah, and that's actually part of the reason why I wanted to end on the inflammation component, because the importance of clearance is something that we know. People who are deficient in MUC5B, there's actually a few papers now showing that there's mutations where people have loss of MUC5B function. They replicate our knockout mouse quite well, which is that they get pneumonia and get very, very sick. So it's something that we need to figure out how to find that sweet spot. So improving but not eliminating the mucus function is something that is crucial here. And I think that's where picking apart one molecule at a time may be beneficial. So if we know like a MUC5AC knockout mouse can live fat and happy in a cage for a couple of years, MUC5B mice can't. So if we can target MUC5AC assembly or its glycosylation under specific circumstances, those would be examples of where we could play with selectivity there.
Brilliant. Thank you very much. So I think that's probably the time for questions now. So we shall thank Dr. Evans for a fantastic talk and question and answer session. I'll move on to our next speaker, who's my pleasure.
Thank you. If I can just have one more comment, I'd like to also acknowledge your colleague, Dave Thornton, as a collaborator on these projects. So we've done a lot of work with the University of Manchester. So thank you.
Right, no, Dave, well, yeah. So it's my pleasure to introduce our next speaker, Dr. Burton Dickey. So Dr. Dickey is a professor of pulmonary medicine at the University of Texas MD Anderson Cancer Center. For the past 25 years, his research has focused on airway mucin secretion, and his laboratory uses genetic approach, knocking out genes in mice and overexpressing genes in mice in airway secretory cells to study their function, and using these genetically modified mice in models of pathologic challenge. So I'd like to hand over to Dr. Dickey for the next talk. Thank you, Vic.
Thank you very much. Good morning, everybody. I think it's worthwhile to pause for just a moment and reflect on how it is that we come to be talking about mucus plugs so much these days. You know, the central role of mucus plugs in asthma pathogenesis, as Chris mentioned, has been known for a very long time, about 150 years, by these German and French pathologists, and then confirmed in the 20th and 21st centuries. But this fact was largely ignored by clinicians until very recently. And I think what changed the field is the imaging in vivo of plugs on CT scan. Prior to this, clinicians had no real way of diagnosing the presence of plugs, and still don't have much in the way of treatment. So it was largely ignored, but I think now the focus is where it needs to be on this very important part of asthma. Chris has given you a great introduction into the structure and the production of mucins, drawing on his own work and that of others. I should also mention that early on, Chris helped us launch our program in secretion, and secretion is what I'm going to focus on. Just that one step in the whole mucin cycle. I do want to acknowledge those who came before me, particularly Bill Davis, and who was in this field for about a decade before we got into it. And what's shown here is a canine trachea, which is laid open. And he's using high-speed video microscopy and looking at these individual mucin granules, and he can watch them pop out one at a time. And they do this spontaneously, and that reflects the baseline rate of mucin secretion that supports mucociliary clearance. Bill then added ATP to the perfusate, and you can see he does it at time 0. And here's the baseline rate of secretion, which he's recorded for many minutes. And you can see that there's a very sudden and enormous increase in the rate of the secretion of these granules. And there's nearly a 2,000-fold increase in the rate of secretion. So this work is fundamental in terms of identifying two different rates of secretion, a slow, steady baseline rate and a high stimulated rate. And that will play out for the rest of this talk. Now, this slide really is the essence of my entire talk. Which is that if we're going to think about mucus plugging in the context of asthma, there are two very distinct processes that are differently regulated, and both are required in order to cause a plug. So this set of images captures all of that. On the top row is Alcian blue PAS staining of the airway epithelium, and you can see that in the healthy epithelium with secretory cells here, you can't even demonstrate any mucin staining. We know that the mucins are there. For Chris first showed that with immunohistochemical staining, which is far more sensitive. MUC5B is expressed in the healthy mouse airway in these secretory cells, but there's so little that you don't see it in a less sensitive histochemical stain. Ciliated cells are, of course, interspersed among the secretory cells. In an ovalbumin allergic model, there is the accumulation of a tremendous number of mucin granules. It looks like the entire epithelium has turned into goblet cells. In fact, that is not the case. Chris showed early on that the actual proportions of ciliated and secretory cells don't change, that there's no proliferation during this process. That's not to say with prolonged asthma, there isn't some degree of hyperplasia in humans, but really metaplasia in which mucin production is being geared up under the influence mostly of IL-13 is what this process is all about. If you then, in this model of allergic asthma, give an ATP aerosol, sort of the complement of Bill Davis's experiment, very rapidly, as quickly as we can fix the lungs, the vast majority of mucin granules are released. And what you see in the bottom panel, stained a little bit differently, and here the mucin is fluorescent red instead of purple, using a stain that Chris developed early on. But here is at lower magnification that airway, not demonstrating any intracellular mucin with metaplasia. You can see that the cells are packed full of mucin. However, there's no mucus occlusion. There's no plug. And it isn't until the ATP is given, the mucin is released, that the plug suddenly forms. Now, it's not that obvious why secretion would cause plugging. After all, you have exactly the same amount of mucin in this image or this situation as you do here. It's just moved from inside the cell to outside the cell. Why does it completely plug the airway? And in order to answer that question, we have to consider three factors: the biophysical properties of mucus, the rapid kinetics of stimulated secretion, and the massive mucin stores. And hopefully in the next three slides, you'll understand exactly how this happens mechanistically. Now, Chris mentioned that mucins are hygroscopic because most of their mass is sugar, which loves water. And in healthy airway mucus, it's important to recognize that mucins are a very, very small fraction of the total mass, less than 1%. So by definition, a gel is a dilute polymer, and in the case of mucus, it's extremely dilute. So in order to get from the interior of the granule, where the mucins are packed dry, into the extracellular space where they need to absorb water, they swell 300-fold. So this enormous swelling largely explains why the mucins then go on to occlude the airway. What you see here is this is a hagfish, which lives at the bottom of the ocean and uses the release of mucins as a defense. And basically, after being stimulated, release a small amount of powder, maybe a teaspoon worth, and it can almost completely turn this bucket into a gel. So I think that that's the most important property to keep in mind in terms of thinking about mucus. And of course, in cystic fibrosis, the entry of water into the airway lumen is deficient as the primary problem. In asthma, it's more a problem of mucin overproduction. But this property of being hygroscopic and swelling tremendously, I think really is fundamental to understanding how occlusion occurs. Now, if the mucus is not fully hydrated, and the concentration's a little higher, say 0.6% or maybe even 1%, the mucus becomes excessively viscous and excessively elastic or firm. In addition, when the mucus is underhydrated, it becomes adhesive. Instead of being a lubricant, it becomes adhesive, and this reversal in properties is used in nature by chameleons when they catch insects. So a second factor is that the rate of secretion of mucins and the swelling of mucins is phenomenally fast. So there's very little opportunity for water to cross the epithelium if there is insufficient water in the airway lumen and large amounts of mucin are released. So this again is Bill Davis's slide showing thousands-fold increase in the rate of release with ATP stimulation. Bill did this with very high-speed video microscopy, and these granules completely disappeared in a fraction of a second, leading him to conclude that once the fusion pore opened, the actual exocytic release of the mucins occurred within a fraction of a second. More recently, this group has shown that the swelling of the mucin granules, they labeled them with acridine orange, occurs within about 1 second. So all the action happens very, very fast and can lead to this. The last thing I want to mention is that I don't think we fully appreciate always how enormous the mucin stores are in asthma. And this is a single mouse secretory, airway secretory cell. And you can see there are hundreds of granules here. The green and the orange are MUC5AC and MUC5B stained with different antibodies. And you can see that both mucins are packed into a single granule. But these granules go from the lumen, the apex of the cell, all the way down to the nucleus. And again, you know, there's hundreds of granules. And in a human airway epithelial cell, the situation's even more dire. These cells can be as tall as 50 microns. And again, they have granules from top to bottom. Now, we have some recent evidence that these granules, which you would imagine would be difficult to reach the surface of the cell within seconds, as Bill Davis demonstrated, and that instead what happens is a process of compound exocytosis, which is illustrated here on the right. And what happens is in the primary exocytic event, the granules closest to the cell surface fuse with the plasma membrane, and then the more interior granules fuse with those. So rather than the granules moving to the surface, they simply fuse sequentially with their neighbors, and sort of the surface comes to meet them. But the bottom line is these enormous stores of dehydrated mucin are made available to the airway lumen and to water and salt very, very rapidly. And the mucins pop out in what's been termed a jack-in-the-box phenomenon. Now, just in terms of sort of intellectual satisfaction, then rather than anything particularly useful, I find it very disturbing to think that the airway so efficiently blocks itself, and imagine that there must have been a very powerful force in evolution that selected for this ability, particularly when you put the occlusion by mucus together with contraction of smooth muscle and close the airway. And I think the evidence is very extensive that this response evolved to block helminths that enter through the skin and then migrate through the lungs and want to crawl up the airway and be swallowed and complete their life cycle in the gut. The airway instead traps them with the mucus and the contraction, and then hopefully kills them. And here's an eosinophil doing its part down here. We weren't the first to come up with this idea, but I illustrated it in 2007. And then Richard Grensis, Dave Thornton, and Chris Evans in a series of two or three papers in JX Med provided experimental support for this by using Chris's knockout mice and showing that fewer helminth were trapped in the airway and more made it down to the gut. What has happened, of course, is that as human beings, we're not challenged by this so much in industrialized societies, but we have allergens that have epitopes that mimic epitopes in a helminth that trigger this response, chitin being one of them, but many other factors with which you're probably familiar. So I think a response that evolved to block the helminths from ascending is instead being triggered from the airway side. Anyway, we've spent most of our time for the past 25 years trying to understand the molecular mechanism, and I'll share that with you. If you're familiar with snare proteins, which are involved in every step of vesicle traffic, three of them come together to make a 4-helix bundle. And one of these proteins contains 2 helices, and that's how we go from 3 proteins to a 4-helix bundle. And that's the core of the machinery. This 4-helix bundle assembles on a platform, a chaperone platform of Monk 18. And the full assembly is promoted by second messengers. And I mentioned the ATP, but the acetylcholine is another very important one that signal through heptahelical receptors to GQ phospholipase C, generating the second messenger's diacylglycerol, and IP3, which act on Monk 13 and synaptotagmin for the final coiling. We've worked out this machinery through a mouse genetic approach, and I'll show you one example, which is of synaptotagmin 2, which is the calcium sensor in secretion. And we begin by confirming expression, and we show it here. We had knocked beta-galactosidase into the SIT2 locus, so we have staining here in the secretory cells and not in the ciliated cells. And what we do is we make the airways mucus metaplastic with an ovalbumin model, then stimulate with ATP. And what you see here is that these mice in which synaptotagmin 2 is deleted fail to release. So we implicate that protein in this process. In that way, we have built up a picture of 6 core proteins that together constitute the calcium-stimulated core exocytic machine. Again, 3 snares, 1 scaffold, and 2 regulators. In a recent paper, Our collaborator, Axel Brunger, biochemically reconstituted this process. And in the interests of time, I won't take you through the details. But again, we come back to the notion that this process can be either genetically or biochemically dissected. Comes down to these 6 core proteins. Of course, there's other participants that help position the granule and whatnot, but these are the core exocytic components. Now, nothing in the airway is that simple. And it turns out that there is a second parallel machine. So amazingly, and for reasons that I don't understand, stimulated exocytosis that we've just talked about uses a different machine from the baseline secretion. Munc18B is the predominant scaffold in stimulated secretion. This is work of Anna Maria Jaramillo when she was a graduate student in my lab. And Munc18A, a close ortholog, functions predominantly in baseline. We have evidence for other components of this baseline machine, such as syntaxin-1, but some of the others are candidates that we're studying currently, but we believe there's an entire parallel machine. Now, the presence of these 2 machines, one of which causes all the trouble, and the other which is responsible for homeostatic mucociliary clearance, affords the possibility of disrupting the stimulated machine. And here we've done that, Anna Maria's done that by deleting Munc18B and showing that she blocks occlusion and airway resistance. I'm going to end here in the interests of time by showing that we've targeted the stimulated machine in a second way by looking at synaptogmin-2, which we consider a better molecular target. And this again was published in that same manuscript. This is the genetic targeting for proof of principle. And then we used a synthetic stapled peptide to do the same thing pharmacologically. My last slide will be to talk about the role of eosinophils in the formation of mucus plugs. And many of you are probably aware that eosinophils are capable of releasing IL-13 and IL-1 alpha and beta, which are powerful stimuli that increase mucin production. In terms of stimulated mucin secretion, of course, they contain ATP that can be released. But substance P acting on neurokinin receptors is something that eosinophils can release, and that directly causes mucin release. They also release proteases that can act on PAR2 receptors. And then indirectly, they interact with neurons to release acetylcholine and can activate mast cells that release histamine, all of which can cause mucin secretion. And then there are eosinophil products that can affect mucus properties, particularly reactive oxygen that I believe John will talk about next. And then the possibility of releasing DNA as part of a trap phenomenon. And I'll stop there and hope that I've left enough time for a few questions.
Thank you so much, Dr. Dickey. I'm so glad you ended up with DNA traps, because that's exactly what we are interested at our end, and we're finding some really fascinating data. My question to you, and in the interest of time, is that you mentioned ATP, and as I understand, and as my colleague Dr. Imran Satya, who is a chronic cough expert, has kind of educated me on the importance of ATP in chronic cough. Where do you think mucus, ATP, and chronic cough kind of intertwine together? And could there be mucus associated with patients who have bouts of chronic cough beyond asthma or COPD? So I'm talking about.
I think that excessive mucus in the airways is a very underappreciated cause of cough. Patients will often identify a tickle in their throat, and they assume that the mucus is coming down from above. Everybody knows about gravity, and mucus does drain downwards. What patients don't know about is that mucus can also travel against gravity, and as a matter of fact, does all day long. It is propelled from distal to proximal airways, as you know, all the way up the trachea, and then enters the pharynx, where it's swallowed. We swallow about 30 milliliters of mucus every day. If it's thin, it goes down, and we're not aware of it. But if it's even a little bit thicker, it can give you a tickle in the back of the throat. So whether the sensation that triggers cough is coming from the lower airways or from the pharynx, I think is variable. But I think that people mistakenly fail to attribute chronic cough to mucus in the lower airways. I would endorse your.
Thank you. In the interest of time, we'll go to the next speaker. Dr. Dickey, if you can please answer any questions on the Q&A in your time. Thank you so much. Our final speaker is Professor John Fahy. He's a professor of medicine at the University of California, San Francisco. And I believe I'm very eager to hear his talk, and I'm sure most of our attendees are. So John, please take it away.
Sure. Well, I want to thank the organizers for asking me to speak. I'm going to talk about eosinophils in asthma with an emphasis on fatal asthma. Couple of disclosures. I am a founder of a company that's working on a mucolytic drug, and I also do some advising to companies, especially in the asthma type 2 inflammation space. I'm going to talk initially just about eosinophils and airway mucus plugs and chronic severe asthma, more to set the stage for talking about eosinophils and maybe neutrophils in acute severe asthma or fatal asthma. I'm going to make a point at the end that maybe chronic plugging in asthma is a result of acute plugs that form during exacerbations, and that the mechanism of persistence of plugs is incomplete resolution of acutely formed plugs. So I won't belabor some of this data because some of it goes back several years now, but this really was an important paper from Eleanor Dunnigan from my lab, where Eleanor came up with a plan and a protocol to quantify mucus plugs in the airway by taking advantage of the bronchopulmonary segment anatomy of the lung, and there's 20 segments in the lung. And we had noticed that we could see mucus plugs on the CT scans as indicated by these yellow arrows here. And this was a little surprising because prior to this, I'd been told before I kind of got into imaging that in order to see mucus plugs, you needed hyperpolarized gas or some sort of non-traditional or non-standard method for detecting them. But these are just regular CT scans, and you can detect the mucus plugging because the airways that are plugged are relatively large segmental and subsegmental airways visible to the CT scanner. And when you look at the scoring, then you see in healthy versus asthma, and this is the Severe Asthma Research Program, you see a bright signal from mucus plugs, where some patients have quite a number of plugs occluding their airways, as many as 10 or 15 segments. And what was interesting about this is that many of these patients didn't complain of cough and sputum production. So to echo something that I think was said earlier, I think our discipline of respiratory medicine has been held back a little bit by an over-reliance on symptom traits that are very insensitive and somewhat non-specific for mucus plugging of the airways. And when we looked at these patients who had high amounts of mucus plugs in their airways, so there's different categories, high, medium, sort of low, you can see that there's a dose response, if you will. The patients that have a lot of mucus plugs have a lot of eosinophils in their sputum. So this mucus plug phenotype associates pretty strongly with an eosinophilic phenotype in asthma. And we could look at cytokine gene expression because we had sputum cell RNA. And not surprisingly, when we see the sputum eosinophils, we see a signal for IL-5 as well. So these patients who are plugged tend to be eosinophilic. And interestingly, then, when Monica Tang in the lab went on to look at sequential scans in these patients, because SARP, the Severe Asthma Research Program, it's a longitudinal cohort study. It has scans done at baseline and then 3 years later. We could see in these scans that patients who had a plug at baseline when they were characterized continued to have that plug 3 years later. And again, this was very surprising because we had been raised as pulmonologists really to think that mucus plugs or mucus in the airways was mobile. And those of us who do bronchoscopy can see that mobile mucus in very mucus forward phenotypes like CF, where there's a lot of mobile mucus in the airway. And that mobile mucus tends to translate into cough and productive cough. But there's this other phenotype where you have these subsegmental and segmental airways that are plugged with these relatively discrete lesions that are not mobile and are actually persistent and stay in the same airway over time. And really, this is quite a puzzle. Why would they not cough it up? I think partly because these airways are beyond the cough receptor network, and there's something about this that is a persistent phenotype. And when you look at the susceptibility to plugging at the participant level, patients who have plugs at baseline tend to have them on this Sankey plot 3 years later, and patients who don't have them 3 years later. So it's not like every asthmatic takes their turn to get a mucus plug. You have it, and then you persistently have it. And not only that, you persistently have it in the same segment. So these 164 patients multiplied by 20 gets you to 3,280 segments. It's 20 segments in the lung. And if a segment is plugged, it tends to stay plugged. And if it's not plugged, it tends to stay unplugged. So there's something about this plug in this segment that is there persistently because presumably there's a susceptibility in that segment for a plug to form there, and then it stays there. My best guess for that is that susceptibility may be driven not only by a type 2 immune predilection, but also maybe because of some prior insult that happened in that airway, maybe a prior exacerbation, maybe a prior viral infection, and maybe there's epigenetic change in the epithelium that results in a kind of a susceptibility factor that's enduring. Whatever the answer is, it's a question that as lung scientists and airway biologists, we need to understand this, because when you understand it, you have some chance of treating it. Now, Monica also did a study where we measured sputum eosinophil peroxidase in these samples. And as I mentioned, there's a 3-year follow-up in SARP, which is nice. And there's also a healthy control group. And if you measure eosinophil peroxidase in these patients, and if you take a healthy group and establish a normal range, that normal range comes out at about 32 nanograms. And about 53% of asthmatics at baseline have high sputum eosinophil peroxidase. And if you look at them over time, approximately the same fraction are high. So you have this persistently low sputum EPX group. That's another important thing to remember about asthma, is about half of asthmatics at least are not eosinophilic. And if you look at the Sankey plot again, you've got these persistently EPX high people. And some of them are very high, like they were persistently up around 1,000 nanograms. And if you look at those persistently high people and you look at their mucus plug score, the blue line here represents these high patients. They have mucus plug scores that are very high and stay very high. Whereas if you don't have EPX, you tend to not have mucus plugs. So there's this association that's not obviously causal necessarily, but the longitudinal nature of the association makes causality a bit more plausible. So, when Eleanor and I published this paper some years ago, when we found this eosinophil association, we'd had an interest in the fact that qualitative changes in mucin polymers, which we'd shown in a prior paper from the lab, could have effects on the mucus gel behavior that were as important as quantitative changes. Again, coming back to how we're sort of raised as pulmonary doctors, we're very much thinking about mucus hypersecretion being a function of mucin hypersecretion. And therefore, the fix is to down-tune mucin hypersecretion. And I think one of the things my lab has tried to do is just really emphasize qualitative changes in the mucin polymer mix. And this is brought home when you think about latex, for example, which is a liquid or at least a soft gel in its native form, but you can cross-link it and it becomes hard without a change in the latex polymer concentration. Similar with fibrin. Fibrin clots form because of cross-linking that doesn't rely on an increase in fibrin polymer concentration. So what might cross-link mucus to make it more plug-like? And peroxidases can catalyze halides or pseudohalides like thiocyanate in the presence of hydrogen peroxide to create these oxidant acids like hypothiocyanous acid, which can then attack cysteines to make them cysteines or cross-linked cysteines. And back to what Chris was saying earlier, it's a very logical target to go after these disulfide links as a rational strategy for liquefying something that's solid. There's other potential mechanisms that could mediate eosinophil-driven mucus plugs. There was a very nice paper from Burton et al. Science 2019 looking at a role for galectin 10 or CLC crystals. So eosinophils, this association of eosinophilia with mucus plugging, there's several biologically plausible mechanisms through which that can happen. So just in terms of summarizing a fair bit of work in this area, if you look at mucus plug high asthma, there's also, of course, mucus plug high COPD. In general, mucus plugs go with more severe disease. It's a feature of severity. So if you want to find a patient with mucus plugging, choose a more severe patient, you're more likely to find the plugging there. It occurs with older age, which is interesting, and it may be back again. If you have a mucus plug score of 19, maybe that's a time stamp of all the mucus plugs that formed that didn't resolve. And so that's logically then going to be a function of age, right? You'll have more plugs if you live longer to get the plugs. I think in some of the pre-webinar questions, there was a question about whether nasal polyps associated with mucus plugging, and we do find as many as 30% of patients who are mucus plug high have nasal polyps. That's a phenomenally high percentage, given that nasal polyps is a slightly odd trait to have. And most, the average percentage in an asthma cohort is about 1%. So it's a very high increase. So again, it's probably type 2 niche biology that's at play here. And these patients, when they've plugged, they have air trapping, they become they're exacerbation prone, whether that's chicken or egg, I don't know. And they have this systemic eosinophilia in addition to airway eosinophilia. Also have high FeNO. Importantly, when we look at COPD patients that have mucus plugs, a high percentage of COPD patients, and it's not the percentage, it's not an overlap with patients with COPD who have asthma. So you can form these mucus plugs without eosinophils. So it's not private eosinophil inflammation. So what about acute severe fatal asthma? Well, Burton mentioned earlier that it's been known for over 100 years, some very elegant pathology studies indicating that when patients die from fatal asthma, they're not dying from concentric smooth muscle contraction and asphyxic, respiratory failure. I don't think you can actually kill a mammal by constricting their airway smooth muscle. We could have a little argument about that, but it's very hard to do it. The best way to do it, if you want to kill someone from an airway disease, have concentric smooth muscle contraction on top of an airway that's already partially occluded with mucus, and then you will kill them. And if you do it across multiple airway branches at a time, you cause death. And this is a nice paper that's a little bit underappreciated in our field, I think, from Boser et al. From 2005. It has a fractal geometry kind of title, which may put some people off because most of us can't think in terms of fractal science, but they did a very nice thing here where they did negative pressure silicone casting, and they had healthy lungs. Well, patients died, but they didn't have lung disease. And you can see the nice silicone cast going all the way out, indicating the tapering of the airway tree. And in asthma patients, I mean, this tapering is completely blunted. And you can see if that amount of airway is occluded and you can't get down to the gas exchanging region, that's an indicator of why people die. And 30% of patients who die from fatal asthma die outside the hospital. And even when you get them in the hospital, you have to be careful not to let them die in the hospital. Some work we did back in the day to look at some of this initially, Claudia Ordonez and Ann Innes did this work when they were with me in the lab, is we took some patients who were intubated with asthma and we looked at eosinophils and neutrophils because for a while in the 80s and 90s, there was this sense that acute asthma exacerbations were eosinophilic storms. There were some sort of type 2 hypersensitivity reaction, some sort of modified anaphylactic reaction. And it became clear from work by multiple people, including Seb Johnson's group in London, that viral exacerbations were the primary driver of asthma exacerbations. And these weren't typically people being exposed to cats. There were people being exposed to rhinovirus. And that we had published a paper supporting that concept by showing that a lot of asthmatics that come to the ED, emergency department, have neutrophils. And here again, in the bottom here, when you take an asthmatic who's intubated, you find a lot of neutrophils in their airways when you just aspirate the endotracheal tube. And we were expecting that neutrophilia to go down as the patient got better, but actually it went up as the patient got better. And some of that's understandable because these patients get a lot of corticosteroids, and corticosteroids will decrease neutrophil apoptosis and increase margination and things like that. But we weren't sure we'd see that in the airway, and we saw it in the airway. There's a lot of neutrophils, even as they get better. Asthmatics, this is 100% on this. You're up to 25% here. In general, we could see some eosinophilia in the asthmatics, and it was tending to go down as they were extubated. So, if you were just looking at this cross-section, you wonder, oh, because the eosinophils are going down, maybe the asthma is getting better because the eosinophils going away, but then why are the neutrophils going up? And the neutrophils, in general were high, I think, because of viral infections again, and the steroid effect here. But then we also began to wonder, well, what's turning over these mucus plugs when they form? Like when patients get better, why are they getting better? Because their problem is mucus plugging. And we did some nice work, and Dave Thornton was involved in this work as well, where we did rate zonal centrifugation and other things to look at the mucus and how it was becoming more fluid over time. And just this is the figure at the end of the paper. We became aware that plasma proteins were also a big part of these plugs, and they were providing a shield against protease-induced digestion of the mucins. And that it was only when the vascular leak settled down and the neutrophil protease amount went up sufficiently to lyse the plug that, this was all sort of association work now, but we've developed a theory that the neutrophils were important in scrubbing the plugs from the airway. And we'll come back to that later on, because our field, I think, needs to think about, like, once mucus plugs form, how and why do they go away? Because that can inform a lot of things, including treatment. So next we moved on last year, or a couple of years ago now, we published a paper just earlier this year, and this is work done by Maud Lijois in the lab, and in collaboration with Tilly Hackett's lab up at UBC. And this was a paper where we took advantage of the Jim Hogg repository of fatal asthma lung sections. And we had a disease control with COPD. We had non-fatal asthma patients who died with asthma, but not because of asthma. We had 5 lungs, 25 plugs. The fatal asthma, we had 9 lungs, 39 mucus plugged airways. And we wanted to really emphasize the plugs, like what was the characteristics of the plugs in these airways. And for that, we did imaging mass cytometry, a panel of 37 antibodies. We looked at immune cells, macrophages, lymphocytes, structural cells, and also we looked at mucins and some other markers as well. And you can see that the beauty of imaging mass cytometry is it really allows you to see the landscape of the airway well, and it can not only show you the wall, but it can show you the lumen. A lot of prior studies have not emphasized the mucus plug itself, so we were kind of focused on our analysis here, both on the airway wall and on the mucus plug itself. And so some of the questions we had were like, why are some plugs infiltrated with immune cells and others are not? Because we could see in the H&E sections of these plugs that some of them looked very acellular and some of them looked very cellular. And we were interested too, when we saw this intense inflammatory look, what was the driver of that? What are these immune cells? And are eosinophils in these cells? And so one of the things we did see when we looked in the wall, we did see, a signal compared to disease-free lungs. We saw some eosinophil heat here. But in terms of the plugged airways versus the unplugged airways, we found similar amounts of eosinophils in the walls of plugged airways and non-plugged airways. So that there may be some susceptibility to an airway plugging because it's got eosinophilic or type 2 immune responses, but we didn't see a big difference between unplugged and plugged airways. Do eosinophils infiltrate the plugs? This is the imaging mass cytometry. You can see there's a lot of cells in the airway lumen, and a lot of them light up for immune cells. And one of the interesting findings was that most, if not all, a very large percentage of these cells, these immune cells, were what we called granulocytes. And we call them granulocytes because they were positive for both ELA2, which is neutrophil elastase, and EPX, or eosinophil peroxidase. So these cells were dual positive for neutrophils and eosinophils. And we were very puzzled about that. Why would they be dual positive? Still not sure we know the answer to that, but we didn't see a huge number of other cells. We did see some macrophages. You can see here on the key, and this is published now, the number of cells that were just single positive for eosinophils was just a small sliver. Single positive for neutrophils was a little bit of a bigger sliver. So the biggest dominant cell here were these dual positive cells. Maud went back and did immunohistochemistry, just got out of the imaging mass cytometry environment to kind of confirm that using these same antibodies, we could see eosinophil peroxidase and neutrophil elastase. And in fact, again, there was this dual positive group. There was some single positive neutrophils, some single eosinophil positive, but most of them were dual positive for both. And that prompted Maud to do some work in an eosinophil airway epithelial cell culture model, where she grew cells at air-liquid interface, exposed it to IL-13 to develop a nice mucus layer, put on blood eosinophils. And then she found that when she looked at the effect of mucus on the eosinophils, it tended to cause the cells to die, but they were not in X and 5. So this was non-apoptotic cell death, suggesting that the cells had undergone cytolysis, which is a non-apoptotic form of eosinophil death, and also eosinophil degranulation. And when you depleted the eosinophils and then exposed the eosinophils, or depleted the mucus and then exposed the eosinophils to the cells, the number of non-apoptotic dead cells went down, suggesting a role for the high molecular weight mucins here. This is just further data to show that. I guess I've repeated that there. So the mechanism of that we didn't fully figure out, but we did take some clues and some hints from the literature around how fibrin can cause cytolysis, and other experimental details from the literature led us to look at CD11b, which is an integrin. When we used an antibody against CD11b, we could decrease this cytolytic effect. When we took away the glycan coat of the mucins that Chris talked about using periodate, we could also decrease it. So there's a glycan and this integrin dependency. You can see here in the normal control situation, the eosinophils detected here by EPX are surrounded by this mucin. When you use the anti-CD11b, or you take away the glycans, you sort of free up the mucins. So there's something about or free up the eosinophils. There's something about the eosinophils being exposed to mucins that causes cytolysis. So what then explains the dual positive granulocytes? It's possible that the eosinophils cytolyse in a mucin-rich environment, and that the neutrophils ingest the eosinophil granules. But again, these dual positive cells are appearing in these chronic plugs, and why this persists over time is really quite puzzling. So why are some plugs infiltrated with immune cells and others not? And one thought we have is the immune cell infiltration may be a function of acutely formed versus persistent, because approximately half the plugs were paucigranulocytic, and the other half were very granulocytic, and in general, again, with these dual positive cells. So some of them are essentially acellular. And what are the features of these acellular ones versus these cellular ones? Well, a big difference is that the mucus plugs in fatal asthma are generally acellular or paucigranulocytic. 66% of the fatal asthma cases had these kind of plugs, whereas 78% of the non-fatal asthma cases had these kind of plugs. So what we're thinking is that these acute mucin-rich plugs occur because of acute degranulation of MUC5AC from goblet cells and a big old mucinous plug that forms acutely. And maybe these granulocytes come in to scrub it, and then that scrubbing is ineffective in some people, and they just get stuck in this non-resolved state. And again, the paucigranulocytic ones tend to be very MUC5AC rich, whereas the granulocytic ones tend to be less so. I mean, there's still a mix of proteins, but in general, the paucigranulocytic ones have a high MUC5AC. And this high 5AC is considered a bad actor. We know some of that from Chris's MUC5AC knockout work. And so in the acute situation, having a lot of sticky MUC5AC is going to occlude your airways. So in summary, the acute fatal asthma cases are paucigranulocytic MUC5AC rich. chronic non-fatal asthma is granulocytic MUC5B rich. I heard someone liking DNA traps. We also see DNA traps in here too. If you're interested in that, we have a figure on that in the paper. Actually dropped it in here. So one possibility then is that these acute plugs fail to resolve because they, in some instances, like normally this granulocytic infiltration results in protease activity that digests the plug. And then maybe in some situations that's effective and you get resolution. And then in another situation, the exacerbation occurs in a primed airway because it's remodeled. And the resolution process starts off, you know, you have this MUC5AC rich plug, but then something happens where the process is interrupted or not fully effective. And maybe what happens is multiple kind of crosstalk pathways can emerge to explain a niche where the mucins and the granulocytes and the epithelial cells are talking together in a way that promotes longevity. And if you think about that, then disrupting this niche, you know, can rationally be done using mucolytics to break up the mucus, anti-inflammatory drugs that take some of these cells out, cells that target the mucins, drugs that hydrate it. There's just at least a framework for thinking about a rational way for looking at these plugs and how to get rid of them. So in summary, airway eosinophilia is strongly associated with mucus plugs and chronic severe asthma. The situation is more complicated in fatal asthma. Acute mucus plugs occur in an eosinophilic type 2 high airway wall environment, but the mucus plugs themselves are usually relatively acellular, and the resolution of acutely formed plugs may involve plug infiltration with the neutrophils and eosinophils, and incomplete resolution may lead to persistence of plugs that are dual positive for eosinophils and neutrophils. So just, I've mentioned Maud because I emphasized her work so much in this talk, and we couldn't have, I'm not an imaging mass cytometry expert. Maud is, Tilly is. I showed some severe asthma research program data at the beginning and funding here. Thanks everyone for your attention.
Thank you so much, Dr. Fahy. That was very thought-provoking. I would like to ask the first question, taking complete advantage of the fact that I'm co-chair. I believe I've also had a conversation with you on this. Of course, there is this whole concept of etosis, and I can see Dr. Peter Weller and Dr. Shige Ueki being online, who are also experts on etosis. There are a number of things that can lead to etosis, one of them being immunoglobulins, immune complexes, and autoantibodies. So that could be one of the things that's increasing in acute. The second factor, which is the ELA positive, EPX positive granulocytes. Could it be that these are heterogeneous eosinophils? Because there has been a concept of eosinophil heterogeneity that was highly talked about and discussed in our recent biannual symposium. So what is your thought on having heterogeneous eosinophils that possibly express ELA2 and they are the ones who are degranulating?
Yeah, I don't have well-formed thoughts. I'm not an eosinophil biologist. I'm actually taking quite a chance coming to this group and talking about eosinophils in the way that I have. I mean, I'm sure there are people on this call who are coming up with reasons why we're seeing these dual positive cells, and I encourage people to come up with that. I mean, the simple kind of clinician way I was thinking about it was that the eosinophils are touching the mucin, and they're being asked, they're cytolysing, and then the neutrophil is taking up the eosinophil granule. And I think there's some literature to support that. But that could be viewed by some on this call as either non-plausible or simplistic. And I would be hesitant to waffle any more than that. Because
honestly, yeah. I think it is simple. I think we complicate things way too much. And in my opinion, we have some data. In fact, we have tabulated some data that shows that free granules in the airway and the sputum, which is a marker of eosinophil activity and cytolysis, is increased in patients who have more mucus scores. So let's watch out on the space. You're on a lot of our collaborating team grants, so we'd have more space to talk on that. But given today's symposia, I was wondering if we can have some Q&A answered. And Andrew, you want to go ahead with the Q&A? Maybe space for 2?
Sure. So the first question is from Helena Agata. So, John, can you see growth of plugs with exacerbations? In the longitudinal data from Eleanor, can you correlate increased length of plugs with number of exacerbations?
It's a good question, Helena. You know, there's a paper in JCI Insight by Brendan Wang where we looked at the size profile of plugs over 3 years, and the average size change was 0. So there was this extraordinary kind of consistency, or the size stayed the same. They didn't progressively grow. I think what's more likely is that with new exacerbations, you develop a new plug, and that lack of growth, if you will, led me to think that that's another reason for some sort of discrete epithelial length that's susceptible. There's a size of the tube that's susceptible, and the plug stays within that susceptible environment and doesn't elongate. And one reason I've got to think about that too is if you think about pulmonary vascular disease and the formation of blood clots in pulmonary veins as a cause of pulmonary hypertension, in that world, the formation of those clots is non-resolution of acutely formed clots as well. So I think I've regularly gone back to cardiovascular medicine to help guide and frame thinking about why these mucus clots form and persist the way they do.
Great, thank you. So I think we've got time for one more. Manali, should we squeeze one more in?
Yes.
Fantastic. So the last question of the day is from Dan and Kunasami. So do any of the speakers see relevance for their findings? Oh, sorry, no, hold on. It's just, oh no, that's gone. Sorry, that's gone. So we'll then go to Stefan Esnolt. Is there EPX deposition in porcine granulocytic plugs due to eosinophil cytolysis?
I don't think so, because I think we actually had a fairly sensitive assay for EPX outside the cell. You know, it was an immunoassay, and so it wasn't. And I think the acellular plugs were pretty acellular. Now there were some. I mean, nothing's ever black and white, but I don't. If the premise of the question is, are we missing some cellular infiltration because the cells are no longer visible? I don't think that's true, because we used protein immunohistochemistry and didn't see that.
Okay, and I think I know what was Danan's question. Danan asked whether, Dr. Fahy, you have been seeing similar mixed granulocytic infiltration in COPD mucus plugs, if you can answer that.
Yes, we did. And that's where I begun to get kind of uncertain. I really do think that there's some role for eosinophils and neutrophils in both COPD and asthma. And I do think we also have to be a little bit careful when we're relying on sputum measures of inflammation to faithfully represent what's in the plugs, because that comes back to my comments earlier about mobile versus non-mobile mucus. So I think there's complexity there. And it was interesting to me that the cellular profile of the chronic plugs in asthma and COPD were more similar than they were different, which is actually nice, because if we could come up with some treatments, they should work across the board, maybe.
Wow. This is great. I think we can keep on with this discussion. I thank you, John, for accepting and coming and giving this talk. I thank both Dr. Dickey and Dr. Evans as well. And again, thank you, Dr. Fahy, all three speakers. We had an excellent tour de force on mucus biology and where eosinophils may come in. I think and I hope that our trainees and our excellent faculty on IES team will have much to think about. Hopefully see you guys for the biennial symposium, and there will be more to talk about mucus and eosinophils. I thank Sanofi, Regeneron, and my co-chair, Andrew, for this webinar. Again, thank you to the speakers and thank you to the attendees. Have a great day.
Bye-bye.